Future of vaccine development. There are several important diseases for which new vaccines are needed to reduce morbidity and mortality globally, which are likely to have a market in both high-income and low-income countries, including vaccine for Group B Streptococcus, a major cause of neonatal meningitis. RSV and CMV. Group B Streptococcus vaccine are currently in trial of maternal vaccine with the aim of induce maternal antibodies that cross the placenta and protect the newborn passively. RSV causes a lower respiratory tract infection, bronchiolitis in infancy, and is the commonest cause of infant hospitalization in developed countries, and globally one of the leading causes of death in those less than 12 months of age. As many as 60 new RSV vaccine candidates are in development as either maternal vaccine or infant vaccine, or involving immunization with RSV specific non-clonal antibodies that have an extended half-life. A licensed RSV vaccine would have a huge impact on infant health and pediatric hospital admissions. CMV is ubiquitous herpes virus that is responsible for a significant burden of disease in infants. 15 to 20 of congenital infected children develop long-term sequelae. Most importantly, sensorineural hearing loss, and the CMV thus causes more congenital disease than any other single infection agent. A vaccine that effectively prevent congenital infection would provide significant individual and public health benefits. Another major line of development of new vaccine is to combat hospital-acquired infection, particularly with antibiotic-resistant gram-positive bacteria such as the Staphylococcus aureus that are associated with wound infection and intravenous catheter, and the various gram-negative organisms such as Klebsiella species and Pseudomonas aeruginosa. Progress has been slow in this field, and an important consideration will be targeting product to the at-risk patient group before hospital admission or surgery. Perhaps the largest area of growth for vaccine development is for older adults, with few products aimed specifically at this population currently. With the population of older adults set to increase substantially, the proportion of the population who are more than 60 years of age is expected to increase from 12 to 22% by 2050. Prevention of infection in this population should be a public health priority. Efforts to better understanding immunosense and how to improve vaccine response in the oldest adult are a major challenge for immunologists today. Novel technologies. Important challenges to overcome in the following years are genetic diversity, for example, of viruses such as HIV, hepatitis C virus, and influenza. The requirement for a broad immune response, including T cells for protection against disease such as TB and malaria, and the need to swiftly respond to emerging pathogen and outbreak situations. Traditionally, vaccine development takes more than 10 years, but the COVID-19 pandemic has demonstrated the urgency of vaccine technologies that are flexible and facilitate rapid development, production, and upscaling. Novel technologies to combat these hardening will include platforms that allow for improved antigenic delivery and ease and speed of production, application of structural biology and immunological knowledge to aid enhanced antigen design and discovery of better adjuvant to improve immunogenicity. Fortunately, recent advances in immunology, system biology, genomics, and bioinformatics offer great opportunities to improve the understanding of the induction of immune response by vaccine and to transfer vaccine development through increasing rational design. New platforms include viral vector vaccine and nucleic acid-based vaccine, antigen-presenting cells such as Dendritic cell, T-cell based vaccine, and bacterial vector are being explored as well, but are still at early stages of development for use against infectious pathogens, whereas classical whole organism vaccine platform require the cultivation of the pathogen. Next generation viral vector or nucleic acid based vaccine can be constructed using the pathogen genetic sequence only, thereby significantly increasing the speed of development and manufacturing process. Viral vector vaccines are based on a recombinant virus, either replicating or not, in which the genome is altered to express the target pathogen antigen. The presentation of pathogen antigens in combination with stimuli from the viral vector that mimic natural infection leads to the induction of strong humoral and cellular immune response without the need for an adjuvant. A potential disadvantage of viral vector vaccine is the presence of pre-existing immunity when a vector such as human adenovirus is used that commonly causes infection in humans. This can be overcome by using vectors such as Simian adenovirus, against which almost no pre-existing immunity exists in humans. Nucleic acid based vaccine consists of either DNA or RNA encoding the target antigen, which potentially allows for the induction of both humoral and cellular immune response once the encoded antigen are expressed by the vaccine recipient after uptake of the nucleic acid by their cells. A huge advantage of these vaccines is that they are highly versatile and quick and easy to adapt and produce in the case of an emerging pathogen. Indeed, the SARS-CoV-2 messenger RNA-based vaccine, messenger RNA 1273, entered the clinical testing just 2 months after the genetic sequence of SARS-CoV-2 was identified, and the BNT162b2 lipid nanoparticle formulated nucleoside modified RNA vaccine was first SARS-CoV-2 vaccine to be licensed. One of the advantages of this vaccine is that they need to be delivered directly into the cells, which require specific injection devices, electrophoration, or a carrier vehicle, and bring with its a risk of low infection rate and limited immunogenicity. Further, the application of RNA vaccine has been limited by their lack of stability and requirement for the cold chain. Beautiful example of how immunological insight can revolutionize vaccine development is the novel RSV vaccine DSK1. The RS surface fusion protein can exist in either pre-fusion conformation, which facilitate viral entry, or a post-fusion, post-F4, whereas previous vaccine mainly contained the post-F4 insight into the atomic level. Structure of the protein has allowed for stable expression of pre-F protein, leading to strongly enhanced immune response and providing a proof of concept of structure-based vaccine design. In addition to the novel vaccine platform mentioned above, there are ongoing efforts to develop improved methods of antigenic delivery, such as liposome, spherical lipid bilayer, polymeric particle, inorganic particle, outer membrane vesicle, and immunostimulating complexes. These and other methods, such as self-assembling protein nanoparticle, have the potential to optimally enhance and scale the immune response to pathogen against which traditional vaccine approach have proven to be unsuccessful. Furthermore, innovative delivery methods such as microneedle patches are being developed with the potential advantage of improved thermostability, ease of delivery with minimal pain, and safer administration and disposal. An inactivated influenza vaccine delivered by microneedle patch was shown to be well tolerated and immunogenic in phase 1 trial. This might allow for self-administration, although it would be important for professional medical care to be available if there is a risk of severe side effects such as anaphylaxis. Also, many vaccines are developed for COVID-19, the new emerging virus, either